Hey guys, this is a tutorial on how to graph your enzyme activity data. And in this tutorial, we're also going to review how to scale axes and plot data points, and then draw a best fit line as well. So for the lab this week, we were doing enzyme activity, and after doing some of the virtual labs, I gave you a data set, a real data set collected by face-to-face lab, and they were collecting data with peroxidase reacting with hydrogen peroxide and a side reaction of oxidizing guaiacol, which made it turn brown. And so the rate at which that mixture turns brown due to the reaction will you can use the spectrophotometer to record absorbance values. And during that time we had 4 groups working, so there was actually 4 replicates, and they mixed the enzyme with the substrate, which was hydrogen peroxide in the presence of guaiacol at different temperatures. So there was 4 degrees here, 22 degrees, 35 degrees. By the way, this is data from that spreadsheet that you're supposed to download to analyze the data, and at 80 degrees here. And so with 4 groups, there was like 4 replicates. So these are the 4 replicates here, 1 through 4. And this is for the 4 degrees. There's the same thing for 22, 35. So this experiment was run at different temperatures. So each group ran it every 20 seconds, starting at time 0. Every 20 seconds, they recorded absorbance values. So the times there are here on the top: 0, 20, 40, 60, 80, all the way up to 120, which is 2 minutes. And from there, with 4 replicates, you can calculate the mean. So that's what you're going to be graphing. You're going to graph the mean values. This data sheet also has the standard deviation, 1/2 standard deviation, and the high and the low value ranges. And you could, if you wanted to, add error bars here, but I'm not going to require it for this one. So everywhere there's a data point, you could actually graph error bars on there. So let's go through how to scale the axis. Now you're going to graph the absorbance versus the time in this first graph. And this first graph should include 5 lines, 1 for each of the temperatures. So this data right here would be used to make 1 line at 4 degrees. Then the data, the mean values for 20, 22, 35, and so on. I'm just going to show you how to do one for the 4 degrees, and then you would go and redo the 4 degrees yourself, plus the other temperatures as well. So the absorbance is actually a measure of the enzyme activity, because the further the reaction goes, the more brown the solution will turn, because you're going to get more product. The product is oxygen, which then oxidizes the guaiacol. So the guaiacol, the side reaction, is what we actually track, indirectly tracking how fast the reaction has occurred. So we're going to graph basically the absorbance. You always want to title your axes, and absorbance has no units. This is because it's based on logarithms, and logarithms are exponents, which have no units. But that's a whole other math lesson. So we're going to do that versus time, and when there's units, you always indicate time on the with the title. Leave enough space so that you can number the axes or scale the axes. Now, in order to scale the axes, you want to make sure that your graph covers, or that your axis will cover, the entire data, so that your data set and your points don't go off of the space you have. So this is the limited space we have, and there are a total of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 lines. And that's time, and the biggest time we have to fit on there is 120 seconds, okay? So it almost looks like this was too perfect and too good to be true. A lot of times it's not that perfect, but let's say you always want to think about, can I go by ones? If you went by ones, you'd only get up to 12. That's not going to fit 120. Then you start looking at other nice round numbers. Can I go by tens, or so on? And in this case, just by happening to go by 10, we're going to be able to fit all the way to 120. So we don't want to go by ones, because that would go way off of there. You don't want to go by 50s, because 50, 100, your graph would be basically up to right here, and that's a lot of wasted space. Okay, so we are going to scale this going by tens, and you don't always have to number every line, because that can create clutter. So this is 10, that's 20, 30, 40, 50, 60, 70, 80. So I'm gonna number every other line, 100 and then 120. Now for absorbance values, based on what we learned in the prior lab, you start to max out on absorbance at around 2. This has to do with the way there's some mathematical modeling associated with it. It involves logarithms. I'm not going to get into that, but the highest value would be 2. And more importantly, you want to make sure we fit our highest absorbance value from the data we're going to graph. Now remember, you're not just going to graph 4 degrees on this graph, you're going to graph the 22, 35. And so we want to scan the mean line. This mean shows the absorbance values you're going to be graphing. They're basically x and y coordinates. And it seems like 1.003 here. The highest values are going to be here at the end, at the 120. 1.266, about 1.3. Here's 1.352. And then here we drop at 80 degrees, we see a lower number there. So it seems the highest value we need to fit is 1.352. There's no way we're going to get that precise on this graph. So we're saying about 1.4 if we were to round there. Okay, so the idea is, can we fit 1.4? And if we went by ones, 1, 2, our graph would just be 2 right there. There's no way. We want to stretch it out further. So we want to look at smaller units. So instead of going by ones, maybe we should go by 0.1s, and let's see if we can fit that data here. This would be 0.1, 0.2, 0.3, 0.4, 0.5, 0.6, 0.7, 0.8, 0.9. That would be 1.0, 1.1, 1.2, 1.3, 1.4, 1.5, 1.6. It seems that if our highest value is going to be about, in this data set, 1.3, going by 0.1s would work. 1 by 1s won't work because that'll be a whole lot of, your graph would be just way too squished down. So following what was mentioned earlier, we don't have to number every one of these. I'm going to number every 0.5 to reduce clutter. So this is 0.1, 0.2, 0.3, 0.4. Right here, I'm going to label this 0.5, 0.6, 0.7, 0.8, 0.9. This I'm going to label as 1.0, 1.1, 1.2, 1.3, 1.4. This would be 1.5, and so we've scaled our axis. So that would be the strategy you would go about. Anytime you're given a data set, you want to make sure you can stretch out your data as much as you can by scaling your axes appropriately. Of course, if you're graphing on software, they do this sometimes a lot, other times for you automatic. So now that we've scaled the axes, we can start plotting points in the graph. And just kind of like using those t-tables that have your x and your y coordinate, your x coordinate is actually going to be the time, which are times 0 through 120. So this row right here is like your x coordinates, and then you're going to graph the y coordinates as the mean absorbance values, because they indirectly measure how much product was made during that time. So the y is actually going to be your absorbance values. We don't need to arrange the t-table. We can just read along here starting at 0 and then come and get the value off of there. So I'm going to use just a circle there for time 0. 0 seconds, we have 0 absorbance. There has been no reaction at that point. At 20, it's 0.142. So 0.142. So at 20, this would be 0.1, and we're not going to be able to graph to the thousands, but we can go to the hundreds because each one of these is 0.1, and we can estimate in between there. So we're going to round that to 0.14. So at 20, this is 0.1, this is 0.2, in the middle is 0.15, so a little bit below there would be 0.14. Then we come to 40 seconds, and it's 0.253, so we're going to go to 0.25, just a little above it. So it's 0.1, 0.2, and then so 0.2, and then that would be 0.3, 0.25 is going to be right there, right about the middle. You won't be able to go to the thousandths place estimating here on your graph. And then at 60 seconds is 0.387. We're going to go ahead and round that to 0.39. 0.1, 0.2, 0.3, 0.4, 0.39 is right by the 0.4. And you start to see a pattern here that the amount of absorbance is increasing at a regular rate. That's expected. If the conditions are the same, you expect that at whatever speed that enzyme is capable of producing this reaction, we expect that as long as there's enough substrate, we expect there to be a constant increase in the amount of a product being produced as measured by absorbance. Then the next time is 80 seconds, and we got to go to 0.511, so we'll go to 0.51. So this is 0.5, this will be 0.6. 0.51 is just going to be right above the 0.5 line, because that hundredths place is dividing that space into 10 equal parts. And then at 100 is 0.617, so about 0.62. So this is 0.5, 0.6. In the middle is 0.65, 0.62 is maybe about right there. And then the last value is 0.726 or 0.73. So this is 0.5, 0.6, 0.7, this will be 0.8, 0.73 would be just below halfway, about there. And so we have what looks like a fairly nice pattern there for your data points. Now, you're going to end up drawing a best fit line, but what you're going to end up doing is you're going to graph the data points for the other temperatures as well. And so while I pick dots here, you might pick other kinds of symbols for the other dots. So that's how you're going to plot the data points. Remember, you're going to have 4 other sets of points on there, and by the time you're done. Now, in the next segment, we're going to go over how to draw a best fit line and determine the slope from that. So now that we've plotted our data points for the 4-degree data, remember we still have 4 more data sets of points to put down for the other temperatures. This is just for the 4 degrees Celsius. We want to decide how our model should be, and it looks like a straight line, and you don't want to connect the dots because it has to be a best fit line, because what we're trying to do is model this with a simplest model we can get, and the simplest model would be a straight line. And if we were to connect the dots with a straight line, you're going to get different slopes. We want a line that fits as best it can one line that has one slope on it. So there's a couple of considerations here. Sometimes when you're plotting your data points on a graph, a scatter, they call it a scatter plot, your points do make roughly, and I'm just drawing them kind of scattered there. Data in real life is messy, never always perfectly on the line. It looks like it makes a straight line, but if I were just eyeballing, I'm just going to sketch, you always want to use a straight edge, but if I sketch that best fit line there, you see that it does not intercept at the origin. It goes somewhere on the x-axis up there. In other cases, the data obviously goes towards the origin, towards 0. And your best fit line would go through 0. In other cases, you know that your data points should go through 0 because of some theoretical consideration. For example, for us, we know that before we mixed the solutions, we had zeroed the spectrophotometer. I remember zeroing the spectrophotometer in last week's lab. You zero it with the solvent before you add anything with color. And now if the color is changing, then it starts at 0 absorption and then moves up. So we're going to want to force our best fit line to go through 0. Now, in order to do this best fit line, you do need a straight edge, so you need a ruler. And if you don't have a ruler, you can get a paper and fold it up. But I know that my line has to go through 0 because of the theoretical reason. Okay? And so I know that. So I know that when I put my ruler down, I'm going to put it down right over this point. So I'll put the straight edge where it's like right there for the ruler. The question is, where do I need to put it out over here so that that line best fits? So what I'm going to do is with my eyes, so it's an eyeball estimation, you kind of gauge where you think that line should go through. And what I like to do, because you can't see through the ruler unless you have a clear ruler, so I kind of like to kind of see where I want the other part of the ruler to be, and I'm going to want that other part of the ruler to be out here. So I make a little mark that I can erase easily, and that'll guide my ruler. So I'm going to lay my ruler on the 0 origin, and then over here on this little X mark here. Now, I don't have a ruler to place on the screen, but I'm just going to use a straight line function in this drawing software, and I'm going to draw that best fit line. I would have laid my ruler just like this all the way across to that point I made out there, and then with my pencil draw that line. Okay? And I probably wouldn't draw the line this dark all the way out there. Okay, so what I'm going to do is I'm going to make it very thin. This is and then my actual line, I'm going to make a thicker line to the line I've actually drawn. So that's just where my ruler would go. Now with my pencil, I would go in there and then draw along the edge of the ruler, right to about where it gets just to where the end of the data points are. Okay. Right about there. And this little pointed out here was drawn very lightly, you can erase it. So that's how you go about drawing your best fit line. Now remember, you're going to need to draw your data points for the other temperatures, so you might use other symbols. You might use open circles, squares, stars, and so on to get the other points. So you're basically going to have 5 lines on here, best fit lines for the different data, right? So when you plot the next temperature, which I think is 22 degrees Celsius, you're going to plot that data and then draw your best fit line for that one. So with the best fit line now, we can actually figure out what the slope is for that line. And because you're going to have 4 other temperatures plotted on the same graph, you also want to make sure that you identify your lines either with a legend or you can label. So you can come here and label this is a 4 degrees Celsius, or you can determine, I still only have that same line there, here on the bottom, you give yourself a legend, and I've already picked just a solid dot for 4 degrees Celsius, and pick other symbols for your other ones and have a legend instead of labeling each one. So with this one line here, I'm going to show you a review from some algebra class, how to calculate the slope, which is how much your line rises for how much that it runs, right? And so the rise is going to be the change in the y coordinate, and the run is going to be the change in the x. And in order to do this, you're going to need 2 points on your best fit line. Do not pick the actual data points, because from one data point to the next, the slopes are different. We want the slope of our best fit model, that straight line. Okay. Now remember that a change in anything, like change in x, is always going to be the final minus the initial, or in other words, x sub 2 minus x sub 1 for the points. The 2 is for the coordinate for point, your second point, and the 1 is for your first point. And for the y, the change in the y is going to be the coordinate for the second point minus the coordinate for the first point. So you're going to need 2 points somewhere on the line. And I'm going to suggest to do something that makes the calculation real easy. It's very convenient. Remember slope, right? So slope is equal to the rise, which is the change in the y, over the run, the change in the x. That's going to be equal to y sub 2 minus y sub 1 over x sub 2 minus x subscript 1. Okay. Now, if you pick for one of your points, this one right here, because remember we forced the line to go right through 0, 0, then that can be your first point. So this can be your first point, and the coordinates are 0, 0. So that means for your y coordinate, that's 0, and for your x coordinate for the first point is 0. So already you're going to have some value minus 0, another value, like you don't even have to do the subtraction because you've picked 0 as your point. But we do have to pick another point somewhere else on the line. I might suggest picking at least your x coordinate where you know for sure. Okay. So let's say we pick 100, so your second point, and remember it's not that data point that's right there, it's the place on the line that's right about there. Okay, so that place on the line is somewhere between, this is 0.5, 0.6, somewhere between 0.5 and, 0.6 and 0.7. So this is 0.6, and this is 0.6, and this is 0.7. Okay. So it's between 0.6 and 0.7, and I see it's not halfway there, so it's not 0.65, it's not 0.64 or 0.63. I'm going to go with 0.63. Okay. For the y, and the x was 100. Okay. So this is very simple, because when you subtract by 0, you get You're not subtracting anything. So this is going to be 0.63, and this is going to be over 100. And then we got to do the calculations. And so 0.63 divided by 100 gives me a pretty small value. 0. I don't even know why I did that. If I divide by 100, just move the decimal place to the left. So I get 0.0063, and there's actual units. The absorbance doesn't have units, but time has seconds. So this is absorbance units that don't have anything like meters or anything like that per second, right? Because this is absorbance units, and this is the seconds, right? And so what you want to do is you're going to put that slope into a table. Okay. So at 4 degrees, you're going to write your slope in there, and this table is in that document where you're following the procedures for this week. We're going to write 0.0063. Now you're going to use these slopes to create another graph called derivative plot. But for now, you write down all your slopes in there, and you got to do this for your other for the other 4 lines for the other temperatures, right? So you got to plot all of those, and you're going to figure out the slopes. Now, the slope, as was talked about earlier in the lab, the slope of any quantity that's created, any quantity versus a graph of any quantity versus time, is going to be considered a rate, like a speed of whatever the y variable is changing at. Okay. But when that quantity that's changing with time has to do with the reaction associated with enzyme catalyzing the reaction, then the slope in this context has a meaning, and so this would be referred to as the activity of the enzyme. Okay. So this would be enzyme activity is the slope. What is the activity? In this particular experiment, it was 0.0063 absorbance units per second. Okay. Now, we are going to, because you're going to end up graphing this, it's hard to graph that small of numbers, so we're going to do a little trick. And what we're going to do is we're going to move the decimal on all of the values in this column. So every value you calculate here is going to be pretty small, but you're going to move that decimal 3 times to the right. And when you do is 1, 2, 3, it's going to be a value of 6.3. But what did we do? We basically multiply this number by 10 to the negative 3, which would give us back the actual value. So when you graph, you're going to put this on your axis right here. It's not going to be the activity in change in absorbance per second. It's going to be the activity in change in absorbance per second times 10 to the negative 3. That tells us that when we label our axis later, that value 6.3 is really 6.3 times 10 to the negative 3, which is really 0.0063. Okay, so you're going to do that with all of these. You're going to get that small number here, and then we're going to have nicer numbers to graph. Like, it'll be easier to graph 6.3 than 0.0063. Trying to scale the axis would be a challenge for anyone, so keep that in mind. So remember, you're going to have to do this for all of these slopes, and in the next segment, we're going to go over what all of this means and how to do that second graph that you need to include in your report. So here we're going to look at how we determine whether or not temperature has an effect on enzyme activity. Now, if we think back to the description of the experiment that was done by that class, they had test tubes that were set at different temperatures, pairs of them, one test tube with the enzyme and the other one with the substrate, and then they mixed them, and each pair had the same amounts of enzyme and substrate, right? So those things didn't vary. The only thing they varied was the temperature at which those pairs were at, right? So we're asking the question here, does our independent variable here, which is temperature, affect enzyme activity? Now remember, the null hypothesis means no effect, right? So it's quite simple. Temperature does not affect. And in this case, we'd want to be specific. It is peroxidase. Okay? Peroxidase activity. And the alternate hypothesis would be what we expect. Temperature affects peroxidase activity. So that would just be saying the opposite, right? So let's take a look at what we would expect if the null hypothesis was true. Okay. We drew one line here already for our 4 degrees Celsius for that data. If temperature does not affect this, then we would expect to get one line per each line that we would get from the different temperatures, because we're going to graph those separate data. We would expect all of them to have very similar slopes to where it doesn't look like there's a significant difference. That would be, this would be what the null hypothesis would be like. The alternate hypothesis would be that at different temperatures we're going to get different slopes. Remember, this is going to be for the same amount of enzyme and the same amount of substrate. You're going to get different slopes. So this might be at 4 degrees Celsius, then the 22. So this would be what the alternate hypothesis would be like. So the idea is to graph the data, and indeed your graph here, while on the prior graph that we were looking at, we went and drew all of that stuff on there. The graph you're going to submit here for figure 1 would be without all of that sketching on there, and you would have 4 other lines with the data points for the other temperatures here. Okay, so that's the way to look at it, and we can do some sophisticated analyses that include the little error bars for the standard deviation and so on, like we've done in the past. I'm not going to ask you to do that on this one, and the techniques are actually even more advanced than when we're trying to model and compare lines. So we're not going to go there. Just graph the lines and then make a determination. Are there significant differences in the slope or not? So you do still need to plot all of these data, and you still need to come up with the slopes and then move the decimal place 3 times to the right so that you can graph in a more convenient units of times 10 to the negative 3. Okay, so that's the basic idea for that. Now, I'm going to show you how to use that data to make the graphs that they give us in the books for enzyme activity. So these graphs are both in your textbook and in your lab manual that you see there on the right. And these are actually what we call derivative plots. And a derivative is the slope. Okay, and so what we're actually doing here, instead of in the prior graph, we were graphing a measure of the enzyme activity, which was the absorbance, versus time for each individual temperature. But the real independent variable in this experiment was temperature. Okay, and what we were trying to do is see what effect does that have on activity, and the activity was the slope. So that's what these graphs are. So they show a couple of enzymes, one from a bacteria that lives in a really hot environment and one just from a typical enzyme found in humans. And they have here the rate of the reaction. And what is the rate? Remember, it's the slope, right? And then these are the temperatures. Okay. So how are these graphs made? They're made by testing at different temperatures, getting those individual lines, calculating the slopes, and then plotting the slopes as your data. So that would be your derivative plot. So basically, for a derivative plot, you're going to be plotting the slope, because that's what a derivative is, versus your independent variable. Okay. And in this case, this is going to be our activity variable, or slopes, versus temperature. And the activity is really has units. So we'll put we can label this peroxidase activity. And the units are going to be in change in absorbance per second. And remember, it's going to be times 10 to the negative 3. Okay, so that's how we're going to be doing these graphs. Now, if I go back to that other page I was on to get that data table, let me copy it and move it over here. Remember, so once you graph, you calculate all the slopes, and you're going to make a new graph. That graph is called a derivative plot. Okay. And in this case, it's going to be the slope versus the independent variable. So the independent variable is going to be temperature, and it's going to be in degrees Celsius. And then here you're going to have the peroxidase activity and the units, because when we calculated the slopes, the units were absorbance per second on the slopes. The units are going to be absorbance per second, but we're going to graph on a smaller scale. So remember, we're going to convert it 2 times 10 to the negative 3, right? So absorbance per second, and then you're going to write times 10 to the negative 3 because we're going to graph converting it to smaller units. So these are actually smaller units, right? So we did that first calculation is 0.0063 or 6.3 times 10 to the negative 3 absorbance per second. And we're going to use that scale. So remember, you're going to have to calculate the slopes. Once you've plotted on the other graph, you're going to need to write in here the slope at 22 degrees. You're going to have to write the slope at 35 degrees, the slope at 50, and then the slope at 80. And those values are going to be small. They're going to be small decimals. So you're going to basically move the decimal place 3 times to the right to get your slope that's times 10 to the negative 3. Right, so you just, whatever that value is decimal, move it 3 places to the left, and then that's what you're going to graph. Now, roughly, I think probably the highest values you're going to get here, once you do that, is going to be probably not bigger than 15. So you're going to have to scale this axis to fit the largest value on here. So I'm not going to scale it for you. I'm just going to leave it here, but you're going to have to scale it to where you're going to fit somewhere between 12 and 15 because that's probably going to be your biggest slope there. The temperature, you have to scale it to fit. And we got to fit from 4 degrees all the way to 80. We'll go ahead and start 0 here, but if I go by ones, 80 degrees is not going to fit. You got to scale it correctly. You can't be changing in the middle. We can't pick one line 22 and the next one 35. You have to go by some scaled value. Okay? Whenever you're doing line graphs. Okay, this is not a bar graph where you can make whatever you want here. So once we pick a unit to go by and scale it, then we got to keep going by that unit. So we can't go by ones. We can try to go by fives. I don't think that's going to go. And then 5 is kind of hard to estimate the in-between. So 5, let's just see if 5 would fit. 5, 10, 15, 20, 25, 30, 35, 40, 45, 50. It's not going to fit. So let's try 10s. 10, 20, 30, 40, 50, 60, 70, 80 will fit. It's not going to take the whole graph, but at least we're going to get it stretched out as far as we can. So this is 0, 10. I'm gonna skip so I don't get so much clutter based on the size of my graph. I don't know if I write on every one. It's going to be too cluttered in there. We know that's going to be 10 in the middle. 20, then 30, then I write down 40, 50, 60, 70, 80, and we don't have data further than 80. Now we do have this first one here, so if I was to scale this to say to go to 15, 1, 2, 3, 4, 5, if I went by ones, I would be able to fit that. So this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is going to be 10, and it's really 10 times 10 to the negative 3 absorbance units per second. 1, 2, 3, 4. This is 5, 11, 12, 13, 14, and then 15 is up here. Well, here I have the 4 degree one. So at the 4 degrees, this is 10. 4 degrees is way out here close to 0. At 4 degrees, I'm going to plot up here, just a little to the right of the axis here. I'm going to plot 6 point. So this is 5, 6, 6.3 is about right there. And then I'm going to get the other slopes. And the other slopes are going to go up higher. I don't know exactly what the values are. You have to plot the data. But we're going to have one at 22. So this is 20. This is 30. 22 is a little past that line. So you want to make sure you got the point. And it's going to be somewhere here. And then the next one is going to be at 35, which is between 30 and 40. It'll be somewhere over here. And these points are going to go up. And roughly, once you do that, then you can go ahead and connect the dots. On this one, you can connect the dots according to the directions. Or you can draw a smooth line. What you're going to find is at 80, there's going to be a complete crash in that activity. It's going to be way down here. And that's due to what was discussed in the background that I asked you to read, the enzyme denatures. Okay, so you can see how this graph right here looks kind of like the ones we see in the textbooks. And so now you know here that the enzyme activity is actually a slope of the individual lines for the enzymes at those different temperatures. They also did one for pH, so you can test pH as doing this. But this is how you do the derivative plot. We've got to graph the slopes, and for convenience, we're going to change it to the smaller unit so that it's easier to scale that y-axis. This is going to end up being figure 2 that you include in your lab report. And the figures, remember, on the bottom, you label it in the order that the graphs come out in your graph, figure 1, figure 2, and there should be a caption as explained in the guidelines there. So I hope this tutorial has helped you on how to do these plots for your lab report. Let me know if you have any questions.